Hello everyone, welcome to the YouTube channel Gene Editors. I'm Maria Cynthia doing my first MSc in Biotechnology at Krista Jayanti College, Bangalore. I'm going to talk about enzymology of DNA replication in both prokaryotes and eukaryotes. DNA replication is the process of producing two identical replicas from one original DNA molecule. We know that DNA replication takes place in the semi-conservative mode of replication. That is, DNA is made up of two strands and each strand of the original DNA molecule serves as a template for the protection of the complementary strand. This process is called as semi-conservative replication. The point on the DNA template where the whole replication process is initiated is called the origin of replication. In this process of replication, a lot of proteins, enzymes are involved. The important enzymes that are involved in replication are DNA helicase, SSB protein, topoisomerase also known as DNA gyrase, DNA primase, DNA clamp, DNA polymerase and DNA ligase. We will be looking onto these enzymes in brief. DNA helicase. This is the first enzyme to act on the template strand at the initiation of replication. Helicase was discovered in E. coli in the year 1976. It is also known as helicase destabilizing enzyme or the unwinding enzyme. Helicase is involved in the unwinding of the double DNA strand into single strands of DNA. How is this unwinding happening? This is happening by breaking the hydrogen bonds between the nitrogenous pairs. This enzyme requires ATP for its functioning. Helicase opens up the DNA and forms a Y-shaped structure which is called as the replication fork. The different factors associated with the functioning of DNA helicases are given here. DNA A initiator protein, which is encoded by the gene DNA A, it is involved in the melting of the ORI site and exposes two templates of single-stranded DNA strands and acts as a platform for loading the replicative DNA B helicase. And secondly comes the DNA B helicase, which is encoded by the gene DNA B. This is also known as tag 1 in eukaryotes and it is involved in the unwinding of DNA by breaking the hydrogen bonds in between the nucleotides. Helicase loader, also known as DNA C, it is encoded by the gene DNA C. It is involved in helping the DNA B helicase to come and bind at the single-stranded DNA strand. This is also called as tag 2 in eukaryotes. DNA helicase is followed by the action of topoisomerase, single-stranded binding protein, and the enzyme primase. In this image, you can see the Y-shaped replication fork is formed. To this, the helicase is bound on the single strand, topoisomerase, and the single-stranded binding protein is also bound. Next, we have the single-strand binding protein. SSB protein coats the strands of DNA near the replication fork to prevent the single-stranded DNA from winding back into a double strand. This is called as replication protein A in eukaryotes. It also protects the single-stranded DNA from being digested by nucleases and prevent the secondary structure formation. This allows the proper functioning of the other enzymes and this also blocks the binding of the extra loading DNA B helicase. Topoisomerase. This is yet another enzyme which is involved in DNA replication. Topoisomerase is also known as DNA gyrase. It reduces the supercoiling and relaxes the tension that is built up during the DNA unwinding, and this enzyme prevents the DNA breakage. It also reduces the topological stress. There are two classes of topoisomerases, type 1 topoisomerase and type 2 topoisomerase. Next, we'll know about the enzyme DNA primase. DNA primase is an enzyme that is involved in the replication of DNA and is a type of RNA polymerase. Primase catalyzes the synthesis of a short RNA segment called a primer that is complementary to a single-strand DNA template. Now, why is the primer so important? Because only if a primer is present, the DNA polymerase will act on the single strand and will start adding new nucleotides. So, after the elongation, the RNA pieces is removed by a 5' to 3' exonuclease and is refilled by the DNA nucleotides. Polymerase enzyme will start adding nucleotides from the 3' end of the primer and will copy the opposite strand. This way, the primer plays an important role. Now let us learn about DNA clamp. DNA clamp is also known as sliding clamp or beta clamp. It is a protein complex that serves as a processivity promoting factor in DNA replication. It is a component of DNA polymerase III, which is a holoenzyme. This clamp protein binds to the DNA polymerase and prevents this enzyme from dissociating from the template DNA strand. Now, why do we need a DNA clamp for the binding of polymerase? Why can't polymerase directly bind on the DNA strand? This is because the protein-protein interaction between the DNA clamp and the polymerase is much more specific and stronger than the interaction between the polymerase and the DNA template. This is the reason why we need DNA clamp. In prokaryotes, the sliding clamp protein is the beta subunit of polymerase III enzyme. It is a dimer. Whereas in eukaryotes, the sliding clamp protein is the proliferating cell nuclear antigen. It is a component of the delta subunit of DNA polymerase III and it is a trimer. We will talk about the beta subunit and delta subunit when we discuss about DNA polymerase further. DNA polymerase. DNA polymerase are enzymes that synthesize DNA molecules from deoxynucleotides, the building blocks of DNA. During this process, DNA polymerase reads the existing DNA strand to create two new strands that match the existing one. The reaction is catalyzed by DNA polymerase in the addition of deoxynucleotides to a DNA chain by using DNTPs as a substrate. Also performs proofreading and error correction. All the DNA polymerase require a template strand which has to be copied. It also requires a primer which is complementary to the template. In prokaryotes, there are three types of DNA polymerases. They are DNA polymerase 1, 2 and 3. In eukaryotes, there are 6 important DNA polymerases. They are alpha, beta, gamma, delta, epsilon and sigma. The DNA polymerase sigma is expressed only in the bone marrow cells of eukaryotes. First, we look into the prokaryotic DNA polymerases. DNA polymerase 1, it is involved in filling the gaps after removal of RNA primer, DNA repair and removal of RNA primers. It has an exonuclease activity from 5' to 3' direction and 3' to 5' direction. DNA polymerase 2 is involved in DNA repair and has an exonuclease activity from 3' to 5' direction. While DNA polymerase 3, this is the main enzyme of replication. It is involved in replication, proofreading and editing. It has an exonuclease activity from 3' direction to 5' direction. DNA polymerase 1. DNA polymerase 1 is a single polypeptide chain which has around 928 amino acids and a molecular weight of 109 kilodalton. DNA polymerase 1 and RNase H are involved in removing RNA primers in the process of DNA replication. It also fills the resulting gaps by synthesizing DNA beginning at the 3' end of the neighboring Okazaki fragment. Both DNA polymerase 1 and 3 have the ability to proofread and has an exonuclease activity from 3' to 5' direction. DNA polymerase 2. It has a molecular size of 89.9 kilodaltons. It has an exonuclease activity from 3' to 5' direction and it is involved in DNA repair. It does participate in DNA replication while it might not be as fast as DNA polymerase 3. It also is thought to be as the backup to DNA polymerase 3 and it interacts with holoenzyme proteins and have a high level of processivity. DNA polymerase 2 also has functions similar to that of DNA polymerase 1. DNA polymerase 3. It is the main enzyme involved in DNA replication. The mass of this holoenzyme is estimated to be 900 kilodalton. DNA polymerase 3 catalyzes DNA synthesis at considerably high rate than DNA pol 1 and 2. DNA polymerase 3 synthesizes DNA in the 5' to 3' direction beginning at the 3' end of each RNA primer. The DNA polymerase 3 has a complex structure and has 10 subunits. These 10 subunits are divided into 4 major classes. The first subclass is the core enzyme. The core consists of 3 subunits, alpha, epsilon and theta. Alpha is involved in the polymerase activity. Epsilon has 3' to 5' exonuclease activity and proofreading activity. While theta, it acts as a stabilizer for epsilon. The second subclass is the clamp protein. The sliding clamp protein consists of two beta subunits. They keep the DNA polymerase enzyme bound to the DNA. The third subunit is the gamma complex. It consists of 5 subunits: gamma, delta, delta prime, psi and pi. It is also called as the matchmaker DNA, and it helps in loading the sliding clamp protein on the DNA. Gamma binds to ATP, the delta binds to beta, and delta prime stimulates the clamp loading. The pi bridges the psi and gamma. The psi is involved in the removal of primase. In this image, you can see how the different subunits of the polymerase 3 are arranged. The fourth subunit is the tau subunit dimer. It acts as a glue to bind together the two core polymerases. This table shows the comparison between the 3 DNA polymerases of prokaryotes. Now we look into the different eukaryotic DNA polymerases and their function. DNA polymerase alpha, it has a polymerase activity and is involved in the primer synthesis and repair. DNA polymerase beta also has a polymerase activity and is involved in the repair mechanism. DNA gamma has a 3' to 5' exonuclease activity and it is involved in mitochondrial DNA replication. DNA polymerase gamma has polymerase activity and 3' to 5' exonuclease activity. It is involved in lagging strand synthesis and repair. Epsilon has polymerase activity, both 3' to 5' and 5' to 3' exonuclease activity. It is involved in leading strand synthesis, gap filling on lagging strand. Next, we'll know about the last enzyme, DNA ligase. This enzyme is involved in filling the gaps formed between the DNA fragments. They join the DNA strands together by catalyzing the formation of phosphodiester bonds. The mechanism of DNA ligase is to form two covalent phosphodiester bonds between the 3' hydroxyl end of one nucleotide with the 5' phosphate end of another. For the function of DNA ligase, ATP is required. In E. coli and other bacteria, NAD+ supplies the energy, whereas in animals, that is eukaryotes, ATP is involved. DNA ligase is used in both DNA repair and DNA replication. These are my references. Thank you.